Welcome to this webinar presentation given on October 20th, 2018. We're broadcasting live right now. This is Zhittya's presentation on FGF1 to treat Parkinson's disease and other neurodegenerative diseases. A breakthrough therapy? Now that's a question. I am John Lobb, CEO of Wound Healing Biopharmaceuticals, and my company has the exclusive rights to the drugs that are being developed by Zhittya Regenerative Medicine. A little background on myself. I'm a CPA. I've been in the biotech industry for over 12 years. I'm the chairman of the board of advisors for the College of Sciences at the University of Nevada at Las Vegas. I'm also the biotech representative for the Biotechnology Industry Organization for the state of Nevada. I go back to Washington, D.C. once a year, talk to the congressional delegation. But more importantly, I'm very dedicated to this company. And I believe that this company, that what Zhittya has developed will make a difference in so many lives. And that's why I have a history in my family of being active in doing things and creating companies that make a difference. My grandfather founded a company called Southwest Gas. It's a $2 billion company. My father ran it for 25 years, and we had a big impact on Las Vegas. I think that what Zhittya is doing and the help that Wound Healing Biopharmaceuticals can give will make a difference in a number of people's lives. It'll be a breakthrough that's will be incredible. So I'm excited to be here, and I'm excited to be moderating this Q&A today. And so most importantly, what I want to say is that we're taking questions. And so if you have questions, you need to send your questions in by email, and then we'll respond to these questions. Now that email is Vika, V-I-K-A, at zhittyamedicine.com. Now I'm going to spell that out for you. Zhittya, Z-H-I-T-T-Y-A, medicine, M-E-D-I-C-I-N-E.com. So that's, send your questions to Vika at zhittyamedicine.com. And Dan, can you introduce yourself?
Thank you, John. My name is Dan Montano. I'm Chief Executive Officer of Zhittya. I'm 70 years old. I received my Bachelor of Science degree in Business and an MBA in Finance from the University of Southern California. After that, for a few years, I was a professor at 2 different universities in accounting, economics, and finance. From 1968 till 1998, 30 years, I was an investment banker in California and London, starting new companies and taking new companies public, IPOs. I probably started 100 companies as a venture capitalist and early investor, and I probably managed the IPO of over 100 companies. I have been the CEO of several high-tech companies, and in 1998, February, there was an article in the Wall Street Journal talking about German doctors who had used a molecule called FGF1 to trigger the growth of new blood vessels in the human hearts. I knew this was a breakthrough. I knew this was big. I got on an airplane and flew to Germany, and I acquired the rights the next week. That started my 20-year adventure now, working with this molecule, which signals the body to regenerate. I was fortunate enough to find Dr. Jack Jacobs, and we have been on this adventure now for 20 years, and I think we're getting close to being able to get FDA to look at us and hopefully seek approval. I am excited that we have identified the mechanism that the body uses to regenerate, and Dr. Jacobs, I know, will be speaking about that. Thank you all.
Thank you, Dan. Now, Jack, can you introduce yourself?
Sure. Thank you, John and Dan. My name is Jack Jacobs, and I am the Chief Scientific Officer at Zhittya Regenerative Medicine. My background includes a PhD in molecular biology from Washington University in St. Louis. I've done stints in academia, in the pharmaceutical industry at Merck, and for the last 20 years, I've been in the biotech industry, trying to get this drug candidate, our drug candidate, FGF1, approved for use in diseases that are characterized by a lack of blood flow to a specific tissue or organ. And today I will talk about our efforts to develop this drug, FGF1, to treat Parkinson's disease and other neurodegenerative disorders. Thank you, John.
Thank you, Jack. and I think we're ready to get started.
Over the next 30 minutes or so, I want to review with you why we believe our drug, FGF1, which is a natural growth factor in our bodies, may be a breakthrough therapy to treat Parkinson's disease and other neurodegenerative diseases. Now, the next slide shows our hypothesis. We believe that inadequate blood perfusion in the brain initiates neurodegenerative diseases, including Parkinson's disease, leading to neuronal cell dysfunction and death. Now, I'm going to be presenting evidence that backs up our hypothesis. I'm going to talk about new medical studies which have examined in these diseases. There's impaired angiogenesis, which is the growth of new blood vessels. Novel imaging technology has shown that in these neurodegenerative diseases, including Parkinson's, that there are blood perfusion deficits. More recently, deficits have been seen in the blood vessels in these regions affected by the diseases, caused by what's called endothelial cell dysfunction. So we'll talk about that as well. And finally, there's some very recent and exciting work that looks at stem cells in the brain. And these findings have shown that in older people, the stem cells are there as much as in younger people. But in older individuals, these stem cells become less active and do not function as well. We believe that is also happening in neurodegenerative diseases. Okay, this next slide just reminds us how incredibly vascularized the brain is. As you can see here, these vessels support and nourish the billions of neurons which are in our brains. They provide the neurons with oxygen, with glucose, and very importantly, they also remove waste, metabolic waste that all cells in our bodies generate. But in the brain, this is very important because waste such as the beta amyloid protein are removed by the vasculature. So how does the brain maintain this healthy state of its vascular system? What processes are important to maintain the brain's vascular system? Well, probably the most important process is termed angiogenesis, which is the growth of new blood vessels from existing blood vessels. And in this slide, you can see simply the growth of new capillaries here, branching off of existing capillaries. So what this gives us is an increase in number of blood vessels in the brain or other tissue, and an increase in density of those vessels. So this process allows better blood perfusion when tissues become damaged or ischemic. Sorry. Okay, let me show you now what angiogenesis actually looks like. One second. I got a cramp in my leg. All right. We can cut that out, but okay.
No, we want that in.
Like, what was it?
You laughed or smiled.
Okay. Okay, let me show you what angiogenesis actually looks like in a tissue. This next slide shows angiogenesis in the leg of an animal who the leg muscle has become ischemic or damaged. When we inject an angiogenic growth factor such as our FGF1 into that tissue, you can see this rich network of new blood vessels which form. So now let's look at the process of angiogenesis. Abnormal angiogenesis leads to many diseases, whether it's excessive angiogenesis or insufficient angiogenesis. Probably the best known example of excessive angiogenesis is cancer. And that is where tumors have to be supplied with blood vessels to grow and divide. And in fact, there are drugs now, anti-cancer drugs, which are on the market that can attack this excessive angiogenesis in cancer. We, however, are going to talk about insufficient angiogenesis in diseases such as stroke, heart disease, diabetic foot ulcers. Insufficient angiogenesis or insufficient blood flow accounts for over 70 diseases in humans and is responsible for over 50% of deaths worldwide. So let's talk about angiogenesis now, and specifically in the brain. This next slide shows that in the brain, the growth of new blood vessels, angiogenesis, is coupled to a second process called neurogenesis, which is the growth of nerves in our bodies from stem cells. So you have these coupled processes, and we know now in diseases that result from ischemia in the heart, diabetic foot ulcers, but also in the brain, that you first must have angiogenesis to stimulate neurogenesis. So we believe in neurodegenerative diseases, as shown here on the left, there's a lack of blood vessel growth and there's a lack of neurogenesis. And that a factor that could stimulate both angiogenesis and neurogenesis, such as FGF1, may have potential to treat Parkinson's disease and other neurodegenerative diseases. Now, I mentioned stem cells earlier. Neurogenesis is the growth of new nerves from stem cells, and some very exciting new work published last April from Columbia University School of Medicine, which is in New York City, showed that in older individuals, the pool of stem cells that they have is the same in number as younger individuals. But in these older individuals, these stem cells are less active. They're not dividing as much. They're not maturing or differentiating into nerve cells. And the reason for this, they postulate, is a lack of blood flow. The same thing that we are postulating is occurring in neurodegenerative diseases. Now, this study from Columbia University School of Medicine generated a lot of interest, and Time Health produced a short video on the study, and I would like to show that to you now. So here's a short video on that study. What FGF1, we believe, can do in the brains of individuals, not only who are aging, but who have these devastating neurodegenerative diseases. Okay, I'm now going to review what that video talked about in this next slide. And this shows neural stem cells, which are in the brains of all of us. And so your stem cells, your neural stem cells, need adequate blood perfusion to divide, so to increase that stem cell pool, but also, more importantly, to mature, differentiate into these other tissues that make up the healthy brain, whether it be neurons or these supporting cells shown here. So in Parkinson's disease, we believe that a lack of blood flow leads to less dopamine-producing cells being produced in the brain, leading to the motor symptoms and other symptoms that are seen in Parkinson's disease. Okay, we've talked about angiogenesis. We've talked about stem cells. Let's talk about a third process which occurs in people as they age and also in these neurodegenerative diseases. And that is a process called endothelial cell dysfunction. This next slide shows a normal blood vessel up here, and endothelial cells are the cells that line each and every one of our blood vessels. These cells are very important in the transport of glucose and oxygen across the blood vessel wall to nourish, say, neurons in the area. And also they're very important to remove metabolic wastes that all cells make to get rid of those toxic metabolic waste. Now let's look at the endothelial cells in someone who has cerebral vessel disease in their brain. This is a disease which affects older individuals. You can see on the left here, normal endothelial cells lining a blood vessel. Well ordered, well packed. In this disease, these endothelial cells become dysfunctional. You can see the endothelial cells here are become distorted. They're not as well ordered. You can even see bumps here in these endothelial cells. So in this blood vessel, perfusion will be limited. So this, again, can contribute to the lack of blood perfusion, which is seen in older individuals as well as individuals with neurodegenerative diseases. Okay, let's summarize now what I've said to date. The neurovascular unit is that area in the brain where the blood supply meets up with the nerves. And if we look here from the entire brain, you can see large arteries give rise to smaller arterioles, which give rise to these numerous small vessels called capillaries. So the capillaries is where the action is, where the interaction with the neuron occurs. You can see right here, this is a capillary lined by, in this case, normal functional endothelial cells. Basically, every neuron in the brain has its own dedicated capillary, which perfuses it with blood, with oxygen, and removes its metabolic waste. And you can imagine if you had endothelial cell dysfunction in this artery, in this capillary, or if the number of capillaries was reduced in this area of the brain, this neuron is not going to function as well. So again, in Parkinson's disease and other neurodegenerative diseases, we feel this lack of blood perfusion in this area is going to lead to dysfunctional, and eventually these neurons will die. Let me show you an actual picture of the brain in an animal that has been damaged by a traumatic brain injury to show you what disordered, dysfunctional microvascular looks like. On the left here, we see healthy capillaries. We're not looking at neurons here, just the capillaries in the brain of an animal. And then afterwards, receiving a traumatic brain injury and then allowing to heal, you see this disordered, this dysfunctional bed of capillaries. One could imagine that a neuron in this setting would not do nearly as well as a neuron in this healthier setting. So again, we believe this localized vascular disorder and decreased blood perfusion initiates Parkinson's disease and other neurodegenerative diseases of the brain. Now in humans, let's turn to humans. You can't go in and be taking slices out of their brain, but some new imaging technology has been developed where in real time you can look at blood perfusion in any part of the brain. This is called functional magnetic resonant imaging, MRI, and this technology has a very exquisite spatial resolution, and it can look down at areas as small as 1/100th of an inch in the brain. And in this study, they looked at the motor region of the brain, that region of the brain which is affected by Parkinson's disease, one of the areas where the dopamine producing cells reside. And looking at blood perfusion in this motor area in a healthy 21-year-old, you see maximal blood perfusion here. As that Columbia University School of Medicine video showed, that as we age, our blood perfusion in the brain goes down, and that's clearly shown here. And remarkably, in Parkinson's disease, there's a 50% decline in blood perfusion in that motor region of the brain. So this goes along with our hypothesis that in Parkinson's, there's less blood perfusion in that specific area of the brain. In patients who have had stroke of that motor region, as expected, you see almost a complete stoppage of blood flow in that area. What about in areas outside of the motor areas of the brain? We know that Parkinson's disease patients have besides motor symptoms, can have their speech affected, can lose their sense of smell. Those are not controlled by the motor regions of the brain, but are controlled by the cerebral cortex, which is shown in this slide. These are the large hemispheres, two hemispheres in our brain of the motor cortex, controlling such things as smell, speech. We know in Parkinson's there's a loss, a slurring or a slowing of speech. And importantly, cognition. Large areas of the cortex here control our executive functioning, our decision making, and our memories. So let's look at how this scanning technology can be looked at to detect perfusion deficits in the cortex area of the brain in a patient with Parkinson's disease. This shows a perfusion MRI scan of the cortex of a subject with Parkinson's and dementia. The blue regions of the scan are normal blood perfusion. Light blue is less, yellow is even less. And these red lesions here show almost a complete lack of blood perfusion in that area of the brain. Now, if you look at these lesions, as we call them, these red lesions, these line up with areas of the brain that are important for cognition, for memories. And so this patient with dementia has a lack of blood perfusion in those areas. One can imagine if you could introduce a drug such as FGF1 to stimulate angiogenesis, to stimulate stem cells in these areas, then you might be able to reverse these symptoms of Parkinson's disease. Okay, I'm now going to turn more to FGF1, talk about it, and talk about its role in stimulating angiogenesis, how it's been used in prior clinical trials in the heart, and then I'll go on and talk about our upcoming clinical trial in Parkinson's disease. Okay, this again shows angiogenesis, the growth of new blood vessels. Occurs, these new blood vessels grow off of existing blood vessels, as shown here. And the critical initiating event in angiogenesis is the stimulation by an angiogenic growth factor, such as FGF1, binding to this receptor right here. This is the initiating event for this whole cascade of events which leads to the growth of new blood vessels. So let's focus on this initial interaction, the interaction of FGF1 with its receptor on the endothelial cell in this case, but these receptors also can exist on neural stem cells as well. But let's look at this initiation of angiogenesis. This next slide shows the FGF1 coming in and binding to its receptor, this Y-shaped structure, which would sit on the outside surface of the endothelial cell. Once that binding occurs, you get this explosion of metabolic events shown here. These processes I won't talk about in detail, but this explosion of these metabolic events, Tanner listed here, lead to the cell proliferation of that endothelial cell, which leads to the formation of those tubules and to the new blood vessels, what you saw on the previous slide. Now, this next slide introduces a very important concept which we and other researchers have found about this FGF1 receptor. This is very important to understand how our drug works in Parkinson's disease and other diseases. It's known that in ischemic or starved tissues, those areas that are underperfused, that the FGF1 receptor is upregulated over time, as shown in this slide. So the left-hand portion of the slide shows normal tissue where the FGF receptor is not upregulated, these Y structures. Then over time in ischemic diseases such as ischemic disease in the heart, we believe ischemic disease in the brain leads to an upregulation of these FGF1 receptors as shown here on the right. So this cell is primed to react to FGF1, but the problem is there's not enough FGF1 around to stimulate that cell to divide and to mature. So in our therapy, we're going to be introducing pharmaceutical amounts of FGF1 to stimulate this process. Let me show you an example in a clinical trial of this FGF1 receptor upregulation in action. This shows FGF1 after injection into the heart of a subject, a patient who had severe coronary artery disease. This is a clinical trial that I designed and supervised, and we injected FGF1 right here. This is the ischemic heart of a patient in the clinical trial. And you can see that after 12 weeks here, we have a blushwork of new capillaries, much as I showed you in the leg of that injured muscle animal, the leg of the injured leg of the animal, where we saw that network of capillaries. So this is angiogenesis in action, mediated by the upregulation of FGF1 receptors. Let's look at an artist's rendition of what we believe is happening, will be happening in our clinical trials. This shows upregulation of FGF1 receptors on neural stem cells. Again, in ischemic parts of the brain, such as in Parkinson's, these would be neural stem cells that could lead to dopamine producing cells. We believe, and I'll talk about how we're going to treat the patients, by introducing FGF1, we will have waves, these waves of FGF1 coming into the brain and seeking out these areas where cells have upregulated FGF1 receptors, as shown here. So here's the FGF1, the yellow balls binding to these upregulated receptors. This is enough to stimulate that cascade of metabolic events, which will lead to these neural stem cells to divide and to mature into dopamine-producing cells in Parkinson's disease. Okay, I want to talk now about our drug candidate, human FGF1. This is a simple protein, as shown on the left here. This was discovered over 30 years ago. I was actually working on that as part of the team that worked on that. And it was first isolated from the brain of animals, and it's only present in very minute quantities in our bodies. So to produce enough of it for clinical trials, we've employed recombinant DNA technology, as is done with a number of other proteins that are in clinical use, and produce this in factories, such as shown here on the right. This is a fermentation factory which can produce unlimited quantities of the FGF1, which we use in the subsequent clinical trials. I'd like to show you some results that we've had with FGF1 in other indications and then move on to our brain indications. As I mentioned earlier, I was involved in supervising a clinical trial where we injected FGF1 into the hearts of patients with severe coronary artery disease. And I want to show you the results of that trial. Rather than talk about the results myself, I'll show you a news clip that ABC News did on this clinical trial. They were able to interview some of the patients at this University of Cincinnati clinical trial site. And I want you to keep in mind, one of the patients that will be interviewed here is a woman named Constance Donnelly. And keep in mind, when you look at her, and we're going to see her angiogram, the picture of the new blood vessels growing in her heart, she received 1/10 of the optimal dose that we gave in that clinical trial. So she was in the low dose group, but she had the most severe form of coronary artery disease. So again, we think this points to the fact that her heart was really starving, was ready to receive FGF1, and she had a very good clinical response. So I'd like to run that video now.
But since receiving an experimental treatment for his blocked arteries, his pain is gone.
I really feel great.
Duke was one of the first heart patients in the country to be treated with a protein actually capable of growing brand new arteries. The genetically engineered protein is injected directly into the heart. Within days, a network of new vessels begins to grow around the blockage, increasing the blood supply. Dr. Lynn Wagner showed us the changes in one patient's heart.
We see a small, narrow main artery and not very many secondary and tertiary arteries. This is after the treatment. What we're now seeing is new blood vessels growing here off the end of this artery.
And the patients themselves?
Symptomatically, they're improved within a couple of weeks of the treatment.
Just ask Constance Donnelly.
Oh, I feel wonderful. I've never felt so good in the last 5 years.
But doctors already see potential in other cases where the blood supply needs a boost, such as strokes and diabetes.
Okay, as Barry Serafin mentioned here, doctors think FGF1 may also have promise in treating things such as stroke. So I want to now move into the brain. I want to talk about some studies that have been done in animals with FGF1 in stroke, in traumatic brain injury, and then I'll turn to evidence, animal evidence why FGF1 may be a good candidate to treat Parkinson's disease. And then we'll move on to talk about our upcoming clinical trial in Parkinson's disease. Let's talk about stroke first. As you know, stroke is caused by a blood clot in an artery leading to the brain or in the brain. And the clot is the stroke area is shown here on the left. There's a core region of dead neurons, and surrounding that core is an area of dysfunctional at-risk neurons. This is called the penumbra. And FGF1 has been shown to decrease the volume of the stroke as well as increase neurogenesis and angiogenesis in animals that have been given experimental stroke. This is shown in this next slide. These animals have been given a stroke. You can see on the left-hand side the infarcted area of the brain, the white area, and then surrounding this white area would be the penumbra, the cells at risk. You see here animals that have been treated with FGF1 show a remarkable decrease in that volume of stroke, and also repopulation of this area here with new nerve cells, which we know come from the stimulation of angiogenesis and then neurogenesis in these animals. Let's look at some other animal experiments. This is looking at FGF1 and its effect in traumatic brain injury. You can see the healthy brain here, and then it's given an injury, and 24 hours later, this is the lesion that appears in the brains of these animals, as shown right here. Treatment for 2 weeks with FGF1, you can see a healing of this lesion. And again, this was shown to be done through the processes of angiogenesis and neurogenesis. Okay, I now would like to turn to the evidence that we believe show that FGF1 is an attractive candidate to test in patients with Parkinson's disease. The earliest experiments looked at the survival of dopamine-producing neurons. We know it's the decline of dopamine-producing neurons in the substantia nigra of the region of the brain, which leads to Parkinson's disease. Early researchers showed that FGF1 was a critical survival factor. They took dopamine-producing neurons and plated them out in Petri dishes, and you can see here, without FGF1, only 25% of the dopamine-producing neurons survived. You add in an appropriate amount of FGF1, you get almost complete survival of those neurons. Now, researchers next looked at animal models of Parkinson's disease. There are two main models, one in rodents, and the gold standard is a Parkinson's disease model in monkeys. Let's look at mice first. In these animals are given a toxin which selectively destroys the dopamine-producing cells in their brains. They come down with Parkinson-like syndromes, all the motor symptoms that are seen in humans. Here you see the dopamine production in the brain of a mouse that's been given this toxin. And you can see when this animal is given FGF1, you get restoration of the dopamine-producing cells in the brain of those animals, and their motor scores improve dramatically. Let's look at experiments that have been done with primates. Okay, these are monkeys that have been given experimental Parkinson's disease with the same toxin as shown here. Over a period of months, you can see their motor scores decline significantly. At this point in time, they are treated with a number of growth factors, including ours, FGF1, and a cousin of FGF1, FGF2, is shown here. What is seen at the end of this experiment is that FGF1 and FGF2 both significantly restore the motor function in these animals, where these other growth factors or a placebo dose did not. Now, FGF2 cannot be developed for reasons I won't go into, But if you look in the brains of these animals at the end of this experiment, as shown on the next slide, you can see that treatment with FGF1 on the bottom here dramatically increases the amount of dopamine and dopamine-producing cells in the brains of these animals. So this is good data to submit to the FDA to back up our belief that FGF1 could be a viable drug candidate to treat Parkinson's disease. Okay, let me just summarize what I've said up to now before we go into the details and design of our clinical trial for Parkinson's disease. We believe if blood perfusion is the problem, and I think the data that I've shown you, we believe it is the problem, that our odds of our success are high in reversing the symptoms of Parkinson's disease. We believe, as I just gone over, that FGF1 will drive three main physiological processes in the brain, namely the stimulation of angiogenesis, neurogenesis from stem cells, and healing of dysfunctional endothelial cells inside the blood vessels in the brain in those areas. Now, we're very lucky that FGF1, unlike other proteins, actually can cross the blood-brain barrier. In animals, up to 4% of a IV or intravenous dose gets into the brain. Here, we're estimating that 1% gets into the brain, and I'll show you some calculations on how much FGF1 we feel will get into the brain with our dosing strategy that we'll use with Parkinson's disease. Okay, let me talk now about our upcoming clinical trial in Parkinson's disease. Let me give you some general comments on this clinical trial. Zhittya is a biotechnology company and will pay all costs for the clinical trials, including transportation. We will seek out clinical investigators. These are neurologists that have experience with movement disorders who are enthusiastic and are hungry to expedite testing of new therapies for Parkinson's disease. The clinical investigator, the woman cardiologist from Cincinnati that was in that ABC News clip, she was not the most famous person in that trial. We had sites at Johns Hopkins, at the Cleveland Clinic. She was our top enroller of heart patients into that trial. She was a hungry clinical investigator, and we will have those types in our Parkinson's disease clinical trial. I want to emphasize that no hospitalization will be required for this upcoming trial. The drug will be administered intravenously, IV, in an outpatient clinic setting. So where are we right now in this process? Before we can do human studies, we have to be sure that our drug is safe by the dosing regimen we've proposed. So we're going to be doing long-term IV dosing to the patients. We had to show in animals, 2 species of animals, that was safe and well tolerated. We've completed those studies. FGF1 is safe and well tolerated at the dosing regimen we'll be doing with our patients. From those studies, we showed that FGF is rapidly cleared from the body by the kidneys. Its half-life in the circulation is only 8 hours. So this is good. You don't want a potent growth factor hanging around the body for a long period of time. There's no accumulation of FGF1 except in those ischemic areas of the body. So where the FGF1 receptor is upregulated, you get a concentration of our drug, and it can reside in those areas for up to 3 to 4 days. Okay, we are now preparing our investigational new drug application, an IND application. Put this all the animal data together, and we'll be submitting that to the FDA by the end of this month. What is the proposed design of our clinical study? I want to emphasize this is what we feel is the best first clinical trial for Parkinson's disease patients. We arrived at this after talking with experts, with neurologists. I want to emphasize there's no guarantee the FDA will agree to this, but we hope they do because we feel this is an optimal trial design. The first study will be a no placebo group. All patients will get the drug. We'll primarily be looking at safety, but we'll also be looking at effectiveness of the drug in reversing Parkinson's disease symptoms. That's done by giving questionnaires and also motor examinations of the patients by the neurologist. We're going to propose to treat the patients, come into the outpatient clinic to get a 2-hour infusion IV infusion 3 times a week. After a month of that treatment, we will do a 1-month follow-up to do lab testing and effectiveness testing. If the drug after that 1 month of administration looks safe, we will then continue with 2 additional months where the patients will get FGF1 3 times a week for 2 months, followed by 1 additional month of follow-up. So a patient's going to have to commit to approximately 5 months of 3 months of treatment, 2 months of follow-up in this study. The total number of sites we will ask for is 4 trial sites. And one thing we'll be negotiating over the next month or 2 with the FDA are what are called inclusion and exclusion criteria. Inclusion is what you have to get into our trial. Exclusion criteria is what, if you have this, you can't be in our trial. And we try to make these criteria as broad as possible. An example of an inclusion criteria is you must be diagnosed with Parkinson's disease. An example of an exclusion criteria is you are pregnant. So if you're pregnant, you cannot be in our trial. We do not know what the effect of FGF1 is on the developing fetus. Again, we'll try to make these criteria as broad as possible to get a diverse Parkinson's disease population into the trial. Okay, I mentioned I did some calculations on how much FGF1 might get into the brain using the dosing regimen, which I just went over. So my math calculations of the doses of FGF1 that get into the brain are shown here. For each 2-hour IV infusion, a patient will get 100 trillion FGF1 molecules into their bloodstream. We estimate 1% of these FGF1 molecules will cross over into the brain, which means 1 trillion FGF1 molecules will get into the person's brain during a 2-hour infusion. Now, work from other groups, not ours, but work from other groups have shown that you need 10,000 of these FGF1 molecules to form what is called an angiogenic foci. So this would be a small region of new blood vessel growth. With that dose, with 1 trillion FGF1 molecules, we calculate that we'll get 100 million foci, angiogenic foci in the brain. And over the entire treatment period, which will be 36 dosing sessions, we will get 3.6 billion angiogenous foci in the brain. So if you look at this figure here in the bottom, these foci occurring here, here, and here, we feel will be sufficient to stimulate angiogenesis and then neurogenesis in the brains of patients with Parkinson's disease. So what do we hope to accomplish with our therapy? This next slide is a hope slide. And this is focusing in on the dopamine-producing cells in Parkinson's disease. We know that in a healthy individual, there are only about 500,000 dopamine-producing cells. And then as we age, these number of the dopamine-producing cells decline. But in Parkinson's disease, as shown here, there's a much more precipitous decline of the dopamine-producing cells, such as when you get down to this region where the patients have lost 60 to 80% of their dopamine-producing cells, you get the motor symptoms characteristic of Parkinson's disease. So in our clinical trial, we'll be dosing patients here. And what we hope through the processes of angiogenesis and neurogenesis, we increase the number of these dopamine-producing cells up into this region, lifting the patient out of this area where symptoms, the motor symptoms of Parkinson's disease display themselves, hoping to lessen or even reverse these symptoms. I want to mention some other clinical trials which we will be initiating. Again, all of these neurological diseases we feel are due to a lack of blood perfusion in the brain, leading to a lack of stimulation of neural stem cells, and also due to dysfunction in the endothelial cells and blood vessels in the brain. These include MSA, multiple systems atrophy, a disease very similar to Parkinson's, but also involving the autonomic system, so patients here have trouble with breathing. Alzheimer's disease, ALS, a motor neuron disease, chronic stroke recovery, traumatic brain injury, and PTSD, post-traumatic stress syndrome. So if you want to find out about the Parkinson's disease trial or these trials, you certainly can go to our website, which is listed here. If you want to talk to someone in our office, you can dial this phone number here. So let me end to say we do not know if our FGF1 is going to work in reversing these neurodegenerative diseases. We have high hopes. As will be mentioned, we're going to try. We're going to try and see what occurs, but we have high hope based on the medical evidence that I've presented to you today. So thank you very much for your attention, and we'll be turning now to some live questions that have been coming in and will come in as we speak.
Well, thank you, Jack. That was great. A lot of good information. And while you're giving your presentation, we got a lot of questions, so I'm ready to start asking you some questions. Jack, our first question is, what is the status of the starting of the clinical trial for Parkinson's disease?
Okay. We hope to start dosing patients in January of this year. And let me just give you the schedule. We'll be submitting our application to do the clinical trial to the FDA, hopefully by the end of this month. There's a 30-day review period. And then we anticipate in December that there'll be some back and forth with the FDA on the design of our trial. So hopefully in January of 2019, we can begin the trial.
Oh, very good. Our second question that came in, how much will I have to pay if I'm in a clinical trial?
You will pay absolutely zero. We will pay all costs for the clinical trial, including transportation if you need to fly or drive to our clinical trial site.
Good questions. Okay, third question. When will I be screened for the clinical trial?
Okay, that's a good question. So in the month of January, when the trial starts, there's a 30-day period where we'll be screening patients. We'll be negotiating, inclusion and exclusion criteria. So we'll be screening you for that. And then we'll be doing some lab analysis to make sure that you qualify to be in our trial. So it'll be about a 2 to 3 week screening period.
Okay. This is a great question. Where are the locations for the clinical trial?
So we're going to ask for up to 4 clinical trial sites in this trial. We know one will definitely be in the Las Vegas area. We do not know yet where the other 3 will be.
Our next question, what are the side effects of the drug?
That is a very good question. And as part of our testing, the FDA makes us test very high doses of our drug in animals. And we push the dose up high, and this is a potent growth factor. So at very high levels, things begin to grow in the body, and the most sensitive organ is the thyroid gland. Now, in our clinical trial, we're going to be about 1/100th of that dose. So we don't expect any side effects in this trial. This drug has been in humans before. It's been in people's hearts, and we've seen absolutely no adverse events at the doses we've tested. So I think I do not expect any side effects at the doses we will be testing.
Good. Okay. Oh, good. We have more questions. That's get this first one here. Okay, Jack, do you really believe that all these brain diseases are caused by a lack of blood flow in the brain?
I do, and others do as well. We don't know what the outcome of this first clinical trial is going to be in Parkinson's disease, but based on the medical evidence, we really believe that a lack of blood flow initiates ischemia in the brain, as I talked about in my presentation, and that by reversing that ischemia, getting blood flow into the brain, we can reverse the symptoms of these diseases.
Very good. Do you think that FGF1 could be useful to treat someone with multiple sclerosis? That's a good question.
That is a good question. Possibly. I think there's a lot of data that multiple sclerosis is an autoimmune disease, so the body's own immune system attacking nerve cells in multiple sclerosis. But there could be inflammation, there could be a lack of blood perfusion that also contributes to that disease. So certainly we would take a look at multiple sclerosis.
I think that'd make people very happy. Thank you, Jack. Third question, another question here. If the growth factor was discovered over 30 years ago, why is it not approved yet for some medical indications? That's
a very good question. As I mentioned in my presentation, drug development is slow and laborious. You know, I think one reason for this is that Merck originally developed this drug and decided not to go forward with it for financial reasons. And because of that, they just basically put it away, put it in the closet. And so Merck sat on FGF1 for many years, and the patent expired on this product. And with that, a lot of pharmaceutical companies and biotech companies stayed away from FGF1. More recently, laws have been passed that drugs that are off patent get some protection once they're approved. So if we get approval for FGF1 to treat Parkinson's disease or any other disease, in the US, we'll have a 10-year period of market exclusivity, and in Europe, it's 12 years. So that's our protection now if we get approval of FGF1.
Well, I think we will, and with your hard work, we'll get it done. And
thank you.
That's all the questions that we have time for right now. There's more questions, but we're going to have to kind of wrap this up. And so I want to thank everybody for watching, and I want to hand this over to Dan Montano, who's going to do our conclusion.
Thank you very much, gentlemen. We want to thank all of you for watching this webinar. We know some of you are watching it from Southeast Asia, from the Middle East. We've had so many people that care about what we're doing because these diseases are worldwide and rampant. We believe we have a treatment for these horrible diseases. If angiogenesis is the problem, we believe we can do something about it. As Jack mentioned, we hope to have at the FDA investigative new drug applications within the next 6 months for Parkinson's disease, multiple systems atrophy, Alzheimer's disease, stroke recovery, ALS, and more. We hope to be dosing people in a few months. Dr. Jacobs, John Lobb, and I will also be in Orlando, Florida on February 7th at the Money Show, where Jack and John and I will be giving a presentation to their VIP Alumni Investment Group. That's Saturday morning. And if anybody is interested in attending, please let us know. We can try and arrange tickets for you. I would also like to tell you that if you wish to attend that, please also email Vika, V-I-K-A, at Zhittya Medicine, which should also be on your screen at this time, and she will try to assist you. In my conclusion, I'd like to make the comment, because a lot of people have asked me, why have I been doing this for 20 years? Common sense tells me if cells in your body are not getting sufficient blood, they're going to suffer. To me, that's common sense. As Dr. Jacobs, Vika, and others on our scientific team keep uncovering that many of these diseases appear to be related to insufficient blood flow. We don't know if we're right. That's what these clinical trials are about. That's what the 20 years of spending a lot of money has been about, is trying to prove that we're right. But what I do know is we won't quit until we have those answers. I want to thank you for your interest. We have a great team of people working at Zhittya, working at Wound, and we appreciate all your support and all your wishes. I personally want to thank all of you who send me emails and call me and tell me that you pray for us. Late at night when I wake up suddenly with a new idea that solves a problem we've been pondering, I hear your prayers. I believe we have a mission, and I thank you for supporting us, and we will continue to keep you informed. Zhittya thanks you, and we will not quit until we know the outcome. Thank you.
Thank you, Dan. Thank you, everybody, for watching.